So let's start with some definitions. Well, everything we see, everything we hear, everything we can feel, touch or smell is a reconstruction of our brain. It doesn't exist really. It's an hallucination. The brain is not like omnipotent. It cannot process everything at the same time. And it has to make a selection for what is important for survival or for the goals that you have in mind. And this selection is what we classically call attention. Now, if we put that into context, let's imagine we're in the middle of the jungle, lost, trying to find our path to civilization. We'll carefully explore our environment in order to collect information and cues, such as, for example, these two shadows in the back of the path on the right. That looks like two people that will drive us back to civilization. So this exploration is serial and slow and goal-oriented, because you're looking for cues to go back to civilization. And if we want to capitalize for every study that have been taking subjects, putting them into an MRI scan to measure increase of blood flow in the brain in order to localize where activity is happening in the brain to perform this kind of task, this is a result that we will have. A dorsal frontoparietal network. So this is a meta-analysis of the past 10 years studies on voluntary control goal-directed attention. Okay? And this network involves the frontal eye field and the superior parietal lobule. Now, we all going to agree that life is not always as peaceful as we wish. And the situation might be slightly different. And often we have to adapt our behavior for survival. In that case, you have to drastically change your system, and your exploration will be more something like that. So you'll be captured by this potential threat. And this is extremely quick, as if your brain was able to process everything in parallel and make a selection onto what has to bypass any strategy. And you'll probably run to the other path on the left. Now, this is really interesting because if you like magic and illusionists, this is actually the trick they use to make you believe that they're doing real magic. They drive your attention or capture your attention on one side while they're popping out some flowers on the other hand. So, for example, if there was like some money on the way, you probably didn't see it. Same idea, if you look at all the studies that have been using functional MRI to find the networks that is classically activated when you have this kind of grab stimulus-driven attention. So if you capitalize all this information, you find that classically you have a slightly different network that is activated that involves a dorsal network, because at the end of the day, it's the orientation of your gaze, but as well as a ventral frontoparietal network, which is now involved when you have to adapt your behavior very quickly and reorient your attention in an automatic way. So I didn't invent that, unfortunately. This is a very famous model that had been introduced by Maurizio Corbetta in 2002. This is summarized here, where you have this control goal-directed attention and this automatically driven attention, both dorsal and ventral frontoparietal networks. Now, I did my PhD on attention, and I went in London to work with Marco Catani to learn more about white matter neuroanatomy. And what he always said to me is, study the behavior, and this is extremely nice, but you need to get your anatomy right. And if you have your anatomy right, you'll be able to build on this neuroanatomy to understand the functioning of the brain. So that's what I tried to do. And the idea was, we have activation in the frontal lobe and activation in the parietal lobe. Is there any connection between those areas? So you start looking in the past and all description of postmortem dissections. Like here you have this nice drawing from Mayo in 1827, where you can guess that you have frontoparietal connections behind like this gray matter here, but the depiction of those fibers was unknown. On the right, you have a monkey brain, and on the other hand, the depiction of the white matter organization in the monkey brain is extremely precise, just based on the fact that you can inject a tracer in the monkey brain, let the tracer follow the path, and then sacrifice the animal and follow slice by slice the tracer. So this is not possible in humans. So that's why we didn't know that much about the organization of white matter. Until we had a new MRI approach that gives us the possibility to peer inside the brain the organization of the white matter. And this approach is called diffusion-weighted imaging. So I'm going to summarize a little bit how it works. So you have here the example of magnified voxels in the cortex and in the white matter. So those tissues are very different. And the MRI scan is going to measure how much water diffuse in those tissues. And it will measure how much is diffused and in which direction it diffuses. So on the left, those are water molecules. They're like diffusing randomly. And the MRI scan is going to record this diffusion. And after like a mathematical treatment, processing, it's going to reconstruct a tensor, which is a model. And here it's a sphere because there is no preferential direction for the diffusion of water inside that voxel. Then on the right here, you are in the white matter. You can already see that you have axons densely packed together. They're going to constrain the diffusion of the water molecule. So this is pretty much what's happening to our water molecules. And the MRI scan is going to record a signal, which after processing is going to look like a tensor like that. An ellipsoid that is deformed according to the main direction of the diffusion of water in that voxel. So the strategy was to do that for the entire brain and piece together voxel after voxel in order to reconstruct white matter pathways. And it's been applied to the perisylvian network, as you can see here, by Marco Catani. So he reconstructed the arcuate fasciculus and found that up to the arcuate fasciculus, you have some white matter fibers that are connecting the frontal to the parietal lobe. And as well as you have posterior connections that connect the temporal lobe to the parietal lobe. It's kind of like a direct and indirect system connecting language areas in the left hemisphere. So I tried to see whether there was some connections above this frontoparietal network, small ventral frontoparietal network. So what I did, I took like the whole frontal lobe and the whole parietal lobe and tried to reconstruct everything that is going from point A to point B. And what we found is, using classic tractography, we only have a ventral frontoparietal network. This was like kind of disappointing because monkeys have like all these connections and we don't. So something here was wrong. So we really asked, like, can we really believe anything that tensor tractography can tell us about the organization of the connections in the human brain? And the truth is, this method is good to reproduce what we know, but when we want to try to discover new connections, you're never 100% sure. And the problem came mostly from the fact that, we have very big voxels with MRI scan. So we're talking about 2 millimeters by 2 millimeters by 2 millimeters in the best case. But we're tracking fibers that we think are between 2 microns and 6 microns. So it's all right as far as they're all going in the same direction, but as far when they start crossing, which at this resolution is almost everywhere in the brain, well, you only see, the dominant population of fibers. So this is what I try to illustrate here. So you have this square here. This is like our little tensor everywhere. And then I magnify this voxel here where you can see that it's a beautiful sphere because you don't have any preferential direction for the diffusion of water in that voxel. And I met with Flavio Dell'Acqua, a physicist who spent his entire PhD trying to solve this problem by applying new mathematical reconstruction to the diffusion signal in order to find where they are crossing and how to track through the crossing of fibers. And his method is spherical deconvolution. So you can see here his face. And this changed drastically the way we have a reconstruction of the signal inside our voxels from a little sphere here to these stars that reconstruct different population of fibers crossing in that voxel. So if you remagnify here, you see that you have like a lot of stars everywhere. And if you do the same tractography, same subject, same areas, suddenly you have much more connection going from the frontal lobe to the parietal lobe. And eventually, if you do that in a lot of subjects and you work out your anatomy, you find out that we're not that different from monkeys. And we also have 3 branches of frontoparietal connections, very similar to the one of our close cousin. So what did I say just before? Can we really believe 100% of what those mathematical reconstructions of white matter can tell us? Especially, can we recreate neuroanatomy based on MRI tractography? Not really. You always have to validate your findings, because if you build on mathematical reconstruction, if you're very permissive, you use algorithms that connect anything, well, you can pretty much carve the connections that you like. So we went to Newcastle to do some postmortem dissections. So we did what we call Klingler dissections, where you take a postmortem brain, you freeze it for a couple of weeks, you unfreeze it, and the water molecules that are inside the brain is going to increase their size slightly when they freeze. And you probably know that if you put a bottle of beer inside the freezer, it will explode. Same idea, but inside the brain. And this will virtually separate big bundles of white matter pathways. So we try to look for the same connection post-mortem, and this is what is illustrated here. So you have like a living human brain on the top, a post-mortem brain at the bottom. This is not the same person. I know, people ask me sometimes, really? So you can see that you can follow those connections on post-mortem neuroanatomy. By then, we could validate our findings with tractography and be a little bit more sure of what we can report. So the next step was to patch it up with the cortex. The cortex is very important as well. So we wanted to reconstruct the projections. And to do that, we extracted the endpoint of every mathematical reconstruction in a group of subjects and just did some statistics. You smooth it and you run a t-test and you find the areas that are the most often visited by the track in the cortex. And this is the result that we obtained. So you have ventral frontoparietal networks that connect the supramarginal gyrus with the inferior frontal gyrus. You have a second branch of the superior longeal fasciculus that connect the angular gyrus to the middle frontal gyrus, and a third branch which is, I started from the lower part, but which is a superior longitudinal fasciculus 1, and it connects the superior parietal lobule to the superior frontal lobe. From then you can start guessing how the brain is functioning. So you have a ventral network, you have a dorsal network. Well, let's say maybe ventral branches for the ventral network, dorsal branch SLF1 is for the ventral branch is for the ventral network, dorsal branch is for the dorsal network, and the SLF2 in the middle is mediating the communication between the ventral and the dorsal network. Just an hypothesis. So one of the features for visuospatial processing and attention is that it is dominant in the right hemisphere. Doesn't mean that it is only happening in the right hemisphere, but most of the time, patients that have a stroke in the right hemisphere recover less than patients that have a stroke in the left hemisphere in terms of visuospatial processing. And the same happens in LC controls. When you want to measure a functional MRI and you're doing a simple post-nap paradigm where you have to detect target that appear either on the left in black or in the right in red, you're going to have an increase of activation in your dorsal and ventral network. What's interesting, and if we want to compare the percentage of increase of activation in the right hemisphere and in the left hemisphere, which in this case are empty boxes for the right hemisphere and full boxes for the left hemisphere, you can clearly see that the percentage of increase is much bigger in the right hemisphere than in the left hemisphere, suggesting a dominance of the right hemisphere for this treatment processing. So our question was, well, if we have a function that is dominant in the right hemisphere versus the left hemisphere, is there an asymmetry in the white matter connections supporting this network? So we looked at the asymmetry for our superior longitudinal fasciculus in the right versus the left hemisphere. And this is what we found. So we found that you have a gradient of lateralization of the frontoparietal connections, with the SLF3, ventral one, being extremely right lateralized, with the SLF2s being slightly right lateralized. So we started thinking, well, maybe we have here the anatomical basis for visuospatial dominance in the right hemisphere. But this is just converging evidence, and what you want is to link it up with the behavior. So we use a standard test, which is called the line bisection, which is classically done in patients with neglect. But if you do it with control, we'll notice that they classically deviate slightly toward the left of the true center. This is true for most, but not all of the population. And this has been always interpreted as a reflection of the dominance of the right hemisphere for visuospatial processing. So very bluntly, I'm going to make an animation of what's happening. This is a real line. This is how I think you perceive the line in your brain, because the visuospatial representation is more important in the control lesional field, the control hemispheric field. And the line that you are bisecting is not the real line, it's the one that you have inside your brain. So you nicely put your mark in the middle, and in real, you've been putting it slightly toward the left of the true center, which is here. So we try to correlate this performance in participants with the lateralization of the frontoparietal connections. And what we found is we had like a unique correlation with the middle branch, the superior longitudinal fasciculus 2. So the more it was right lateralized, the more you were deviating towards the left of the true center. Interestingly, we found that we had also like a few people that deviate slightly toward the right of the true center and have a bigger second branch in the left hemisphere rather than in the right hemisphere. So that was interesting. But you know, when when you're doing MRIology, the real question that you always want to know is like, is there a real anatomical basis behind my findings? And how can I bridge it up with biology? So, you're just like, okay, I've got an asymmetrical volume of my mathematical reconstruction. What does it mean? Why my connection will be bigger in one hemisphere compared to another hemisphere? So there is not like an infinite possibilities. Well, you can have more connections. You have more axons, can take more space. And if you have more connection, you're going to process your information more in parallel, and it's going to be indeed faster to process it. You can also have larger axons. Now, this is an old graph that is showing that according to the diameter of your axons, you're going to increase the conduction velocity. So information is going faster in big axons than in small axons. And finally, you can have more myelin. So we read that in the textbook, and this is the original publication that is showing that myelinated axons are conducting much faster than unmyelinated axons, independently of the fiber diameter. So all that, let us think, well, that's great. We have a spatial distortion on the line bisection, and we can find that it's related to the lateralization of the SLF2. But if that's true, that bigger connections are related to one of these three factors, the processing should be faster in one hemifield compared to the other. So we did like a simplified post-snap paradigm. So for those of you who don't know, you have to fixate the cross. You have a box on the left, box on the right. You have to detect targets that eventually appear on the right or on the left. And on the real post-snap paradigm, you have a queue in the center as well. But this one was simplified. And what we found is that, our participants were slightly faster on the left compared to the right hemifield, but they were responding with the right hand, so it makes completely sense. But this asymmetry still correlated with the lateralization of the second branch of the superior longitudinal fasciculus, here, as you can see. So, in a way, you deviate as much towards the left of the true center, then you are faster to detect target on the left. And this is related to your neuroanatomy and the symmetry of the frontoparietal network. The healthy visuospatial network, in few words, seems to be supported by a frontoparietal network of connection divided into three branches. Those connections, at least for the ventral one, are lateralized to the right. Viability in those connections can predict the performance in healthy controls. And in that case, maybe more philosophical, functions seem to shape or sometimes reshape neuroanatomy. Now I'm going to go on to the dysfunctional frontoparietal network. So during neurosurgery, classical approach is to use electrodes to temporarily inactivate small cortical areas. You can see a human brain here as a neurosurgeon is going to perturbate electrically a small area. This is very valuable because this gives the opportunity to neurosurgeon to map in real time the functional area in the brain and avoid to remove core functional areas and damage the behavior of the patient. So it is a wonderful approach. So we've been working with Hugues Dufault and went to see him with our test, our line bisection, saying, well, we think that, the right hemisphere is important as well. You should try to do some line bisection to avoid to induce neglect in your patient, which he did. So our prediction was the following. You still have your line. This is a real line. If you perturbate the frontoparietal network in the right hemisphere, going to perturbate the representation of the line, which bluntly, I'm going to say it's going to be shorter, but it's probably much more complicated than that. And this is the line that you're going to bisect according to what the line that you have in your brain. But the real line is different, and that's why you deviate towards the right of the true center in that case. So this was our prediction. And the localization for the deviation was the following. So we had small deviation where we were stimulating the posterior part of the superior temporal gyrus. We had small deviation where we were stimulating the supramarginal gyrus. But the real stuff was happening when the neurosurgeon was stimulating inside the white matter. And we had large deviation up to 3 centimeters or 5 centimeters here. So while controlled stimulation didn't induce any significant deviation. Then the neurosurgeon went a little bit further, and he stopped when the patient, even in the absence of stimulation, was showing significant deviation. The good news is 5 days after the neurosurgery, the patient was absolutely fine and not showing any deviation at all at the line bisection and didn't have any visual neglect. So let's talk a little bit about visual neglect. So visual spatial neglect is like a fascinating syndrome. It really looks like the brain is only reconstructing the half of the visual spatial environment. So they've been like impaired and like, truncated from one half of the world. And it's been interpreted as the non-allocation of our limited processing resources to the opposite side of the lesion. In other words, it's been interpreted as patients are not paying attention in the neglected side. So you can see here, as my hands, I'm asking a patient to do this simple neglect task. This is probably the easiest you can do. And so he has to cancel every bar here, and he's only canceling the one that are situated on the right part of the sheet of paper. Well, the video is quite long because I've been very insistent, but it's absolutely not exploring the left side. He's actually sometimes like canceling twice the same bar, looking at me like this is really annoying. So this is an example. You can also assess it using cancellation of letters here. So you have to cancel all the A across like different letters. This is harder, and patients are more likely to show difficulties at this task or asking to do line bisection. And if you do that in patient with a stroke in the right hemisphere, at the acute stage, you have 80% of those patients, or around 80% of those patients, that will show signs of visual neglect. And when you go to the chronic stage after 3 months, well, half of those patients that were showing visual neglect will spontaneously recover, and the other half will not recover. And they will still show visual neglect. So one of the $100 question has well been like, can we predict the one that will recover and the one that will not recover from their stroke? So we can focus our treatment and re-education to the one that are not likely to recover spontaneously. And this is how you're looking for the neuroanatomical basis for chronic visual neglect. So now we've been having a lot of papers about that, including mine, and there's been a huge debate, very angry people. But at the end, maybe we all agree. And if you plot all those results into the same brain here, you get like all these letters, either electrical stimulation studies or lesion overlapping studies. Well, if you plot them all together, you find out that, it's all on a frontoparietal network. I was actually quite pleased about that because this was my hypothesis, like that, if you damage your frontoparietal network, you'll have visuospatial neglect. So, but it opened like another question. Why are you sometimes having a lesion here and visual neglect, or a lesion here with visual neglect, and your analysis is not highlighting the track? It's mostly because we're not used to do statistics for white matter connections. We used to have topological voxel-based approaches. So the idea since Broca has always been we collect a sample of patients showing our pathology Here, visual neglect. Let's simplify the brain in 3 areas, A, B, and C, like 4 patients, all with visual neglect, and you overlap the lesions to find out that actually, well, all the patients have a lesion in area B. Not all the patients have a lesion in area A and C, and that means that, well, it's probably area B that is processing the visuospatial information and damage and leading to visual neglect. Now, as I said, this method didn't seem to give a consensus in the result across authors, because as you can see, some authors reported frontal areas, others temporal areas, and others again parietal areas. So 2/3, 3/3, well, almost all the brain, because sometimes they also report occipital lobe. So we thought like maybe we can change the way we're doing this kind of analysis by using an odological approach. So you have the same model, 3 areas, but you inject knowledge about how those areas are connected. And you do adapted interpretation of the lesion, where lesions are plotted onto this model. And you can see that now they all disconnect area A and C, and area B is not responsible. So those approaches are complementary. There is not one to replace the other. It's something important to keep in mind. So this is a theory, and we tried to put it in application for visual neglect. So we collected 58 patients, similar age. Well, our neglect patient had a stroke a long time ago, and our non-neglect patient was a little bit less time between the stroke and the evaluation, but we can all assume that non-neglect patients are not going to develop neglect later unless they have another stroke. I think it's fair to assume this. Neglect patient, of course, deviated at line bisection and later cancellation. The lesion size was bigger than the non-neglect patient, so every statistic that we carried on was using lesion size as a regressor to remove this potential factor as a predictor. And the method was the following. You're taking your patient brain, bring it into the MNI, and overlap knowledge about the white matter neuroanatomy to quantify the probability of disconnection for every patient. And you can now doing that automatically with the software that we designed, which is free, and you can download on our website, brainconnectivitybehavior.eu. And then we just plotted the results. So in dark gray here, you have the percentage of chance, or the risk, having a disconnection for patient with visual neglect, and in light gray for patient without visual neglect. And you can see that mostly you have differences at the level of the peripheral visual network. They were all like middle cerebral artery patients. We didn't have any posterior cerebral artery patients. And when you cancel out the effect of the lesion size, only one track remains significant. It's the frontoparietal SLF, predicting the chronicity of visual neglect with a sensitivity of 81.6%. So 4 patients out of 5 had a disconnection of the frontoparietal network, and a specificity of 95%, which is only 1 patient had the disconnection and no visual neglect. So we did also like approaches looking at the severity of the task. So here now you have the number of omissions for the later cancellation task. Again, there was a frontoparietal connections. And the same thing for the deviation. So the track, when the track is disconnected, you have more deviation on the line bisection. And then we push the model a little bit further with the 3 branch of the frontoparietal network with a new model for tractography. And it's most likely, again, to be the middle branch that is responsible for visual neglect. So let's get back onto this. So you have a sensitivity of 81.6% and a specificity of 95% for middle cerebral artery stroke, showing visual neglect after a disconnection of the frontoparietal network. 4 out of 5 patients that have a disconnection of the frontoparietal network have a disconnection of the frontoparietal network. So what kind of damage have the patient that don't have a disconnection of the frontoparietal network? Where is located the lesion? So we went back to the original overlapping of lesions for those 7 patients that didn't have a disconnection of the frontoparietal network, and this is what we found. They all had a lesion involving the thalamus, particularly the lateral dorsal nucleus and the pulvina, which have been frequently associated with neglect. So you have cases described in the literature. And if you add on those 2 predictors for middle cerebral artery, you explain 97% of your patients showing chronic visual neglect. So in conclusion for this part, the damaged visuospatial network happens when the right frontoparietal network is disconnected. It leads to visuospatial neglect. So we've seen that. I hope I convinced you. Little is known about the equivalent phenomena in the left hemisphere. It's true, there is a real lack of tractography studies or track studies onto right neglect after a left hemisphere stroke, which happen less often, but still happen quite often. And the relation between the lateralization of the connections and the recovery of visuospatial neglect has not been explored. So, in other words, are we all the same? If you're more likely to have bigger connection in your left hemisphere for the frontoparietal networks than the right hemisphere, are you more likely to recover if you have a stroke in your right hemisphere due to the configuration, your proper configuration of your brain? So we've been speaking about the healthy visuospatial network, and we've been speaking about the damaged visuospatial network. Probably some of you will think, okay, that's interesting, we're speaking about visuospatial, but I'm not working on visuospatial and I still have a frontoparietal involvement. We all specialize on a given function, and we explain a given function, but maybe there is some collinearity between different functions in the frontoparietal network. So we had the innocent idea to try to collect all the literature about the past 10 years reporting frontal and parietal co-activations and do a huge meta-analysis trying to find out, which area is activated for what, and does it belong to a different branch. So we went through all those different functions. You got visuospatial, working memory, language motor, and other functions. They all activate the frontoparietal network. And here we only report the activation in the frontoparietal network. We're not reporting the ones that were in the temporal lobe. Otherwise, it would have taken years to collect the information. So this represents 850 studies. So the first thing that strikes us is those areas are very similar. I remember when I was doing my PhD, the girl, Emmanuelle Vold, she was working on working memory. She said, well, we're working on the same network, but with a different window of understanding. Now I'm going back to it. I realize that we're all working on the same network with a different window of understanding. So we try to quantify how much you can explain these variants, and if there is not a function that'll be collinear to all that, explaining what's going on. So we entered the pattern of activation in a principal component analysis, and we found out that, you have 2 factors that explain about 70% of the variants, which are a dorsal frontoparietal network and a ventral frontoparietal network. And they both overlap into a network which is right in the middle that is systematically activated, whatever you do for the 14 function I showed to you. So that was interesting. And try to explore what are in these two components? Is there like a random separation of those functions or something that makes sense behind that? We'll find out that, the first component is dedicated to any kind of task that is special. And the second component is dedicated to any kind of task that is non-special. And again, the overlap is for every task. Then we look at the contribution of the three branches of the superior fasciculus to these different activation. We found out that, the dorsal one is indeed activated and supported by the superior fasciculus 1. The ventral one is mostly by the superior fasciculus 3, and the common activation are supported by the superior fasciculus 2, that is systematically activated by all these functions. So this network has been discussed by John Duncan for a long time as a multi-domain pattern network. But some other authors also highlighted that frontoparietal network may indeed be involved in a much higher cognitive level of function. Particularly, if you run tasks onto a conscious report of what you see or what you feel or what you hear, and you do a contrast between they consciously report it or they're not able to consciously report it, you're going to have, when you're not conscious, just an activation of the primary area, but when you report it consciously, you have the primary area and the firing up of an entire frontoparietal network. And if you do a big meta-analysis of all those functional MRI studies, find out that actually they all activate branches of the frontoparietal network. Same goes for the measure of metabolism in patients that lose consciousness, either temporarily for slow wave sleep, on anesthesia, or during vegetative states. They show a huge decrease of metabolism in the frontoparietal network. And again, this link up with the connection, not only the connections that we've been describing, but even the definition of visual neglect. So if you wonder, what I'm trying to do from now in Paris with my lab, so we're trying to make a link between those four dimensions, which are stroke, neglect, the definition, the connection that are supporting this network. What is this unique function that is supported inside this network? How does this relate to functional activation when you consciously report something? How does this viability of the functional activation relate to the viability of your connection? As well as how hypometabolism in vegetative states and disconnection can predict recovery or not. I thank you all for your attention. I'll drive your attention to the special issue of Cortex that we're doing with Tim Chalice. Is a single brain model sufficient, for which submission and review are most welcome. Thank you very much.